Hi guys, in this video we're going to be looking at GM and transgenic technologies. We're firstly going to be talking about what GM or genetic modification is, then look at transgenic species and the definition of those, then a way of obtaining a transgenic species through recombinant DNA, followed by an example of transgenic E. coli that produces insulin. So firstly, genetic modification, also called genetic engineering or GM, is where we permanently change the DNA or the genetic makeup of an organism. Now the important thing here is that it's a permanent change that we're making. One example of genetic modification is creating transgenic species. And this involves taking a gene from one organism and putting it into the DNA of another. So we're moving a gene. For that species to then be considered to be transgenic, all the cells within that organism have to have the gene in it, and it needs to be able to be passed along to the offspring. Okay, so it needs to be in all the cells and be passed on so that the offspring also have that gene within them. One way that we can create transgenic species is through the technique of recombinant DNA. And there's a few steps to this. So firstly, identification. We need to work out where the gene is that we want on the genome of whatever organism we're getting that gene from. So this is pretty hard stuff to start with. You've got to actually identify that gene and where it is found on the DNA. And the way that we do this is by using markers and comparing it to known genes. So if we know that there's other genes that do perhaps a similar sort of thing in a similar sort of organism, we can then use that to give us a bit of an idea of where this gene is going to be found on the organism that we're going to take that gene from. Once we have worked out where that gene is, we then need to isolate the gene. And the way that we do this is by using what's called DNA restriction enzymes. And these are enzymes that cut the DNA at known points. And basically, most of these are isolated from bacteria. So they're enzymes that we've taken out of bacteria. And we've got about 800 different ones that we know and can use. So what this means is once we've mapped the DNA, so we've looked at the GATC, whatever, and worked out where that gene is, we then work out the start, what sequence is at the start, so that GATC might be the code that's at the start. Then we look for the enzyme, restriction enzyme, that will cut the DNA when it sees the GATC. We put that enzyme in and it will cut that DNA at the start. There's another restriction enzyme which will cut it at the end, and now we have that chunk of DNA which forms the desired gene that we want. Now, when a gene is transcribed, only the gene that codes for the protein is going to be transcribed. Around this gene, they're going to be regulatory genes, and these are the instructions on when and how that gene is to be used or expressed. So when we're cutting that gene out of the species, that gene that we want, we may need to add other parts to that genome that tell the new organism when to switch on and when to switch off that gene. So we add these regulatory marker genes to our gene that we've already isolated. Now that we've got the gene the section of DNA that we want to put into our new organism, we need to get lots of these. And the way we do this is through what's called a polymerase chain reaction. Basically, it's a machine you put in a whole heap of free nucleotides, and it heats that section of DNA up, dissolving the hydrogen bonds, then cools it down. Those free nucleotides then join on to the exposed base pairs and create double copies of the DNA. So basically, polymerase chain reaction is an artificial way that we simulate DNA replication. It works fairly similarly. Once we've got lots of those sections of DNA or those genes, we then need to insert that gene into our target organism. And there's different ways that you can do this. Some genes and some sections of DNA, you can just use a very, very fine needle and inject them into the nucleus, and they'll work that way. Other ways involve putting that gene into bacteria and then using that bacteria to infect the organism so that all the cells in that organism have the gene in it. And there's also a thing called a gene gun, where you basically shoot that section of DNA into those cells. So different cells and different types of organisms will withstand and or need specific ways to actually get that gene into the genome of the organism. And this is a fairly hit and miss part of this transgenics. An example of a transgenic species is there is a special type of E. coli that has the human insulin gene injected into it. So the first thing they did is isolated the human insulin gene from the human genome. They then added the regulatory gene for beta-galactosidase, and this is an enzyme that tells the human insulin when to turn on and when to be produced, as well as they needed to turn off other enzymes that work within the E. coli, which then would destroy this human insulin around it. So they needed to turn on some genes and turn off other genes. They then inserted that gene into a weakened form of E. coli, a weakened strain of E. coli, and the gene then took in that E. coli. So now we have a transgenic E. coli, so it's an organism, a bacteria, that has a gene from another species, in this case humans, within it, and it passes it on to its offspring, so that we can say that it's a transgenic species. So this E. coli will produce human insulin as it grows, and this insulin can be harvested for medical uses. Prior to this creation of the transgenic E. coli, we harvested insulin from pigs, so diabetics would have to actually inject pig insulin, which is fairly similar but a little bit different to human insulin. So some people did have problems with this. So this is a much better way to harvest that insulin. In this video, we've talked about genetic modification or genetic engineering, being the permanent change of the genome of an organism. We've talked about transgenic species, where you take a gene from one species and put it into another species. We've looked at recombinant DNA as a way to get a transgenic species, being that you firstly identify, then using restriction enzymes cut out this gene or section of DNA that you want. You can then add your regulatory enzymes to it, use the polymerase chain reaction to make lots of copies, and then insert those copies into the desired organism. When these genes take hold, for that organism to then be called a transgenic species, remember, it needs to firstly have that gene in all of its body cells, and secondly be able to pass that gene on to its offspring. And we've looked at an example of a transgenic species being E. coli, or a particular transgenic version of E. coli that produces human insulin as it grows, which can now be harvested for medicine. Thanks for watching, guys. Peace out.